Welcome to Too Big Online School in collaboration with Confidence Publications. All rights reserved, neither complete nor in parts. This tutorial may be duplicated in any form by cut, copy, paste, camera, screen recording or any other means, or incorporated into any electronic or mechanical or digital system without permission. So, topic for the today's lecture is peptide and polypeptide overview. Today we will try to learn the two terms peptide and polypeptide and what is the difference between these two terms. So a peptide consists of two or more amino acids. When two or more amino acids, first amino acid 1 and amino acid 2, A1 I should write, and A2, these are attached together, and A3. This is termed as a peptide. It can be if two amino acids are there, then it will be called dipeptide. If three amino acids, then tripeptide. Then four amino acid tetra. Then five amino acid pentapeptide, and so on. Now, what oligopeptide is? Oligopeptide, when the amino acids are usually between 12 to 20, then it is called an oligopeptide. See, there is no clear-cut boundary of how many amino acids should be in oligopeptide, but usually there are 12 to 20. If you cannot say if there are 21 amino acids, then I will not term it as oligopeptide, or if there are 14 amino acids or 11 amino acids, I will not term it as a oligopeptide, or I will term it peptide. There is no clear-cut boundary, but for the experimental use and general user-friendly use, we use the peptide. So, for example, in the lab, I am going to synthesize the peptide of say 22 amino acids. So I will call it a oligopeptide, and it is by the name oligopeptide, it is clear that it is somewhere in this range only, and anyone will not guess it to be of 100 or so, 100 or more amino acids. And third term is, first term is peptide and oligopeptide, and third is polypeptide. Poly means many. When many amino acids are joined together, it is called polypeptide. So even the polypeptide term does not have a clear cut that how many amino acids should be there, then we will term it as polypeptide. It is usually taken that more than 20 amino acids, I will term it as a polypeptide. A polypeptide may contain as many as 100, even 100 or 200 or something, or even more than that. So technically these all are polymers of amino acids. See, in chemistry there are two terms. One is monomer, the unit which forms a polymer, and there is a polymer. When many monomers are attached together, a monomer, and when many monomers are attached together, it is called a polymer. So it is called a polymer. So technically these all are polymer of amino acid, and because of their different lengths, they are named different. So let us draw a polymer, or rather a peptide or oligopeptide or polypeptide, you can term anything, or a dipeptide. So we are going to draw a dipeptide first of all. So for drawing that, we should know what is the structure of the amino acids. So in amino acids, we know that there is a alpha carbon here. This term is alpha carbon. The carbon attached to the C is known as alpha carbon. So this C, carbon attached to this C is known alpha. If there will be one, then beta, then gamma, and all that. So this is termed as alpha carbon, and alpha carbon has 4 different groups attached to it. One is the H group, other is the NH2 group, other is the COOH group, and fourth is the R group. So as a polymer, what is a polymer in the chemistry? We know that the polymers are formed by the condensation of the monomers. So we will try to draw two polymers, two monomers for 2 amino acids, and then we will try to condense to form a dipeptide. So first of all, let us see this. This is a alpha carbon. It has one H group attached to it, and it has other R group attached to it, and it will have COOH group attached to it, and it will have a NH2 group attached to it. Then similarly we will draw another amino acid. This is the side chain, you know, so we will have one NH2 group here, and COOH group here, and here it will again be H, and here it can this side chain. So we will try to condense these two molecules together to form a dipeptide. So let us first try to expand. If you have studied in chemistry, you can write the detailed structure of COOH group. So I will just try to write COOH in the detail. So from it can be C double bond OH. This is how the COOH group is written. This group COOH. Now I will again expand these NH2 group. I will just erase this, and I will just expand it. So this is 2 hydrogen were attached to the and 2 hydrogen were attached to the N. Now if let me change the color. Now you see that the OH of the COOH and H of the NH2 group, these condense together and water is eliminated out. CHOH. I can write HOH as water or H2O water. You should know the structure of water. So I have just eliminated HOH, and now HOH is eliminated, and I will make a bond for. So let me eliminate this CHOH now eliminated, and H from the NH, and a bond is formed between them. Let me Control Z. A bond is formed between them, and this bond is called peptide bond. Let me type it. Okay, Control Z, and then peptide bond. Okay. Okay. So what it now, this peptide consists of two amino acids. First amino acid this, and second amino acid this. Then it is called dipeptide. Then there similarly can be tripeptide. The COOH of this and an NH2 group of here another of the third amino acid will condense together to form again a peptide bond, and then there will be 3 amino acids in this peptide, and it will be called tripeptide. Suppose first amino acid 1, amino acid 2, this is now dipeptide. Again one more amino acid 3 tripeptide, amino acid 4 tetrapeptide, amino acid 5 pentapeptide. When 12 to 20, then it can be termed as oligopeptide, or it can be of more than till 100 or something, then it will be polypeptide. So technically everything is a polypeptide or polymer. Technically everything is a polypeptide and polymer, but because of their different lengths, we name them different. Again, now examples of some peptides include, spelling mistake is there, it should be peptides. Okay, so examples of some peptides are there: oxytocin, you must have heard this name; amanitin, glutathione. A dipeptide consists of 2 amino acids, is aspartame. Now, a very important thing, you should concentrate here, that this is the alpha carbon, and here also this is the alpha carbon. Sometimes the side group can contain COOH, or side group can contain here NH2. So sometimes the bond can be formed between this COOH and NH2. In that case, it also will be the dipeptide. It will also be a dipeptide, because then where 2 amino acids are condensed together, it is usual that it will be a dipeptide. But this occurs to lesser extent in proteins. Dipeptide, tripeptide can occur of this length, but it is not common to proteins. Okay, one more point to be mentioned here is that if there is suppose there is a series of amino acids attached together. See, here is A1, A2, A3, A4, A5. Now this amino acid will be termed as N-terminal amino acid. Terminal are the end points, no? So why N-terminal amino acid? Why? Because you see in this dipeptide, at the end there is this nitrogen group, and here at the end there is this COOH group. So this will be called N-terminal amino acid, and this will be called C-terminal amino acid. So you can see a polypeptide is a dipole. Dipole in the sense that it has 2 different ends. This end is different from this end. Okay, that's all for this lecture. Thank you for watching as always. Thank you for watching as always.